Okay, so today I'm going to be sectioning a Eudicot bean plant stem. So here we have it here. In later parts of this video, I might section a leaf as well as a root. So I'm going to dip the stem into the water, as well as my flat razor blade. Holding it in between my two fingers like this, I am going to cut a really, really thin piece off. And you might want to do this multiple times if it doesn't work out for you to find like a perfect piece, but for now I'm just going to cut one. Here we have it on the slide, and I'm going to be using a dye called Fast Green, which is at a concentration of 0.1 moles. So here we go. I'm going to just drop one drop of this solution onto the section. I'm going to wait 10 seconds, and then I'm going to wipe it off with a Kim wipe. So it should be 10 seconds. Just gonna blot it right off. Then I'm going to take a cover slip. Oh, before I take the cover slip, I'm actually going to add one drop of glycerol to keep the sample hydrated. So here we go. Okay, there it is. And then, so now you take the cover slip and we put it on at a diagonal angle so that we ensure that no air bubbles get in. So here we have the finished product. I'm going to repeat this process with the leaf and root as well. And I'm just going to look at this under the microscope. Okay, so now I'll be doing the leaf sectioning. Here I have a bean or Eudicot leaf, or true leaf, and I've rolled it up so that it's easier to cut. And I'm going to just cut off the front so that it's all even, and then I'm going to make a really small incision like this. And if it's too thick, I can just restart, get another piece. This one looks good. So now I'll just take tweezers and put it on a microscope slide. Okay, so I'll be using Fast Green to dye the leaf. So I'm going to first just drop one drop of this Fast Green onto the slide, or a couple of drops if needed. I'm going to wait 10 seconds for the stain to just diffuse into the leaf, and then I'm going to wipe it off with a Kim wipe, or suck it all up with a Kim wipe. Okay, so now I'll just take a cover slip, and before I put it on, I'm going to get a drop of glycerol and put it on there so that it stays hydrated and in place. Okay. Now the cover slip. I'm putting it down at an angle so that there's no air bubbles. And then I'll just look at this under the microscope slide.